Hello, I would like to welcome everybody to NIA Artificial Intelligence Lecture Series. Our speaker today is Dr. Papademetris, who is a professor of radiology and biomedical imaging and biomedical engineering at Yale. I met him while working on NIH reviews. He is very enthusiastic about the field and very dedicated to science. His reviews were always among the most comprehensive and thoughtful. His research interests are in the areas of medical image analysis and the creation of software for medical applications. He is the first author of the recent textbook, Introduction to Medical Software, Foundations for Digital Health Devices and Diagnostics. And he will be talking today about lessons from the regulatory process for medical software for image analysis and AI. And I'm turning over to Dr. Papademetris.
Thank you, Dr. Tsap, for the introduction. We were there maybe 4, 5, 6 years ago when the onslaught of AI started hitting the study sections, small business, NIH Small Business Review grants. Dr. Tsap was the SRO of, and I was a reviewer. So this is the title for the talk, Lessons from the Regulatory Process of Medical Software for Image Analysis and AI. These are my disclosures. This doesn't relate to the talk, really. And let me tell you a little bit about what I'm trying to do with this talk. So we have a set of highly promising methods in artificial intelligence machine learning. Some of us are seeing results that are better than anything we have seen. Let me minimize myself here. And that's sort of important. There are some issues that are worrying us, me at least, and others in the field when it comes to the use of these methods, things like nonlinearities, overlearning, adversarial attacks. All of those are related, essentially. And the fundamental question is, how do we learn when to trust these results? We get some really good results, but can we trust them? Are these likely to be still good in the subsequent use? And what I'm going to try to do during this talk is to try to see if we can learn some lessons from the regulatory process for medical devices, medical software, and apply them to AI research and medical image analysis research to help us understand how the process works. Okay, so here's the current situation we're living in. AI, whatever this is, we'll define it in the next slide, has redefined medical image analysis and what has come to be known as software medical devices, or SOM. These are regulated software tools through FDA and other regulatory agencies for use for diagnosing and treating patients. AI is kind of like gene therapy or stem cells, has created wide interest in the general public and fears and reactions. So back in the day, maybe 20, 25 years ago, when the hot thing in image analysis was level sets, nobody really knew what level sets were or cared about that. Everybody's now an expert in AI. If you walk into any medical school, everybody's clearly an expert in AI. And our challenge really when you see results published out there is to separate the hype from the reality. And that's really what we're dealing with. So here's a cartoon, just to add to the hype, drawn with one of those text-to-image synthesizer stable diffusion. And this is a picture of a monkey climbing a tree. And the quote comes from the 60s. This is Stuart Dreyfus, one of the early researchers in this field. And he said, there's lots of hype in AI. And, we go back and forth. He said, it was like claiming that the first monkey that climbed a tree was making progress towards landing on the moon. So yes, we're making progress, but are we ultimately getting there, or is it just an illusion? So just keep the monkey in place. Yeah. So with that, it's very easy to use AI outcomes to generate promising results, but do these things scale? When are these things too good to be true? So that's where we're dealing with as we speak right now. So that's where we are. So the hype didn't start yesterday. You can go back to 1958 in the New York Times, and there's an article about the first perceptron. And here we go. The Navy revealed that the embryo of an electronic computer today, they expect will be able to walk, talk, see, write, reproduce itself, and be conscious of its existence. The embryo, the Weather Bureau's $2 million 704 computer, learned to differentiate between right and left after 50 attempts in the Navy's demonstration for newsmen. The service said that it would use its principle to build the first of its perceptron thinking machines will be able to read and write. It is expected to be finished in about a year at a cost of $100,000. So this was 1958. Clearly, this never happened, right? We know that. Here's hype slide number 2. This is not, we're not going to play these videos, but this is on YouTube. You can find it. This is from Jeff Hinton, one of the fathers of deep learning. And this is the comment: we should stop training radiologists now it is just completely obvious that within 5 years, deep learning is going to do better than radiologists. Now, I sit in a radiology department. We have lots of radiologists. We're having a shortage of radiologists, and deep learning is nowhere near replacing radiologists. So I can tell you this much from my own lived experience here. So what is AI? We've heard the words. Let's just get some definitions. This is from one of the international standards, ISO 22-1989. You get this very, very broad definition. An artificial intelligence system, this is an engineered system that generates outputs such as contents, forecasts, recommendations, or decisions for a given set of human-defined objectives. So this is basically almost everything. Outside expert circles, my working definition, just talking to people at the medical school here, is that AI is some magic that allows a computer to do something. And lots of traditional software tasks are now seen as AI. So people will call AI just about anything. Okay, so if it's done by a computer, it's AI. So let's just back up and think a little bit about a taxonomy here. What does it take to convince a computer to do something? There are sort of two main ways. There's an explicit way where we describe all the rules in detail. This is traditional software programming. And here we have human design to a great level of detail and computer execution. And then we have implicit methods. We have no rules or even understanding sometimes by human. We have lots of input-output examples. When you see this, do that type thing, the computer uses learning processes to create a procedure that mimics the examples. And you can say that the human designs the learning procedure and the computer then figures out the code. Old-fashioned AI is explicit, things like your favorite expert systems when you used to call your bank to ask for help and say press 2, then press 3, then press 4. Machine learning falls into the category of implicit systems where we just give it examples, and everything from like the simplest regression model to a deep neural network comes under that category. Here are some of the challenges of using these implicit methods, and this is for primarily machine learning in imaging, although the same things apply to other things. What we learn is super specific. An algorithm used to classify pediatric brain tumors may be useless for adult brain tumors. That's just one example. An algorithm works on 3 Tesla MRI as a field strength, that's the most common in hospitals, may be useless on 4 Tesla MRI. An algorithm that works on a particular resolution may fail badly at a slightly different resolution. We have examples of that if you want to see them one day. And what we do is that we solve one very specific problem at a time. There are no guarantees that this method will work for any other problem. And we often do not understand how it works, and we're not able to predict how it fails. So this is our challenge. Something works really well when it does, but it's super specific, and we don't know how it will fail. And this is what sets the stage for the talk. We have these methods that often significantly outperform standard approaches. How do we learn when to trust them? This is a fundamental question. And what I'm going to do is that I'm going to look at the medical regulatory process for software to get some inspiration for some ideas as to how to go about that. So for those of you who are not super familiar with the regulatory process for medical devices, medical software, here's a common misconception. This is how I thought things worked 10 years ago, maybe. You code your software however you like. At the end, you perform a validation study. You submit the results to the FDA, and if the results are good enough, you get approval, and then you're free to sell and make money and make your fortune. This is not how it works. The way it works is that you start out in your company and you establish a quality process for creating software. You have a lot of rules in place for who can code, what training they need, how do we go about committing code, etc. We design our software and we document it. We perform risk analysis to figure out what could go wrong. The FDA will optionally review your plans ahead of time. You implement your software and document. You perform a pre-specified validation study. You submit the entire process to the FDA. The testing is part of it. You get approval or clearance. These are technical terms. We're not going to get into them now. And you're still responsible for monitoring how the whole thing works after you've sold your product. So it's a much more involved process. And the first step is this thing called the quality process. So the medical software process is all about establishing trust. And we want to ensure that devices are safe and effective prior to being marketed for medical use. If we're thinking about medical image analysis research, we want to ensure that our algorithms are safe and effective for whatever task we're using them for. There was a paradigm shift in industry, maybe sort of this dates about World War II, and this is the shift from quality by testing to quality by design. And this is sort of, we build it however we build it, and we make sure it works at the end, to having processes that ensure that what comes out at the end is good every time. And these ideas came to full development in post-World War II Japan during the process of reconstruction. And they were important in the United States and the West more generally in the late 70s, early 80s. You've heard of things like total quality management, the Toyota way, all of that is the sort of core of what quality systems are. And this led to the creation of modern quality systems, which is now embedded in the medical device regulations beginning in the mid-90s, probably. So what do these things include? This is one of the regulatory guidance documents from one of the international organizations, International Medical Device Regulators Forum. Again, the goal is to have quality by design, not just by checking at the end. And it comes out of recognition that testing, while valuable, is not sufficient. Okay, you can't just test quality into something. We cannot trust the results unless we have a good process in place. Maybe we got this due to pure luck. Maybe our algorithm worked really well because we found images from patients that were really not particularly variable and we would like it. There are explicit procedures on how key things are done. And this includes, and this again, another lesson for our research population, training for personnel. You can't just have somebody in a lab, barely knows what they're doing, download some code, download some data, and come up with results, and you feel like, yeah, that was good results that are publishable. People need to be trained to be qualified to do the work they're doing. Otherwise, we risk getting random results. Sometimes they work. The other thing, and if you remember one thing from the talk is probably this slide, there's a concept in the regulatory process of risk classification. So the focus is on what the software will be used for, not on what it does. And these are the so-called use cases. So once you know what the software is going to be used for, we estimate the risk involved. How bad would it be if the software or device failed? How likely is it will fail? Can we live with the failure? And based on the use case and the risk involved, we end up with a risk classification, something like low, medium, high. There's a million of these schemes. They're all slightly different, but they all boil down to something like this. And the same algorithm can have different risk classifications depending on the use case. So it's not what the algorithm does, it's not what the tool does, but how it will be used that drives your risk classification. So let's look at some practical applications. On the right, you'll see a table of AI-enabled medical devices comes from the FDA's own web pages. And most of these tools that are cleared for human use are intended as human assist devices, for example, computer-aided diagnosis. And the reason for this is that it lowers the risk level because there's a human in the loop. If you're just dealing with not fully automated things, the risk is lower because the idea is that a human can catch the error. Algorithmically, these are identical often to what could automated diagnosis tools. You could use the same tool for human assist or completely automated diagnosis, but the risk profile is very different because you say, well, we have an expert that's going to look at the results, so we're a lower risk tool. And that's why actually most of these tools tend to be, designed this way. What is the practical impact of the classification on the nitty-gritty of creating software? AI tools as part of that. If you have higher risk, you require more detailed documentation when you submit to the FDA, for example, a detailed software architecture design. And on the testing side, if you have higher risk, you require, for example, unit testing and more detailed clinical testing. So the scrutiny goes up as the risk level goes up. And that is the principle of the thing here. What are the implications for some of our own research outside of the medical device process? We should expose things that are more impactful, that make more impactful claims to higher scrutiny. If this is a claim that this is going to massively affect everybody, and it's a high impact paper, we should put high scrutiny on it. And we should always keep in mind, again, and you'll see examples of failure later in this talk, that the change of use case of research requires a new evaluation. Here's an obvious example. We do not use lab studies to get drugs approved. They do clinical trials, even though theoretically it's the same process, it's the same drug. The lab paper, the research paper from the university, the use case is to demonstrate, initial feasibility. You cannot use that to get clear and approval for putting a drug into the population at large. And I would argue that we should not rely on AI or other software-driven results from papers for major public policy decisions without additional testing. And we saw that during the pandemic, all these models that were published and then people were using them to make decisions, were those subjected to the level of scrutiny that you would have taken for that use? So we'll come back to that at the end. So with that out of the way, we'll look at machine learning a little more detail to establish some common ground for everybody. We'll identify the two key problems in my mind, the possibility of overlearning and the issue of nonlinearity. And this undergird a lot of what you will see, domain shift and other terms that you will have heard. And we're going to look again at ideas from the regulatory process for how to address a little bit of those as well. So the most common AI task is probably classification, is labeling something as good or bad, as healthy or disease, what have you. And the typical process goes, given some measurements, descriptors or features, we assign a label to these measurements. And this is often a two-step process. Given a thing, a human being maybe, you map it into measurements of features. We call this representation perhaps. So a person could become the two numbers, height and weight. And given these measurements, the features we classify. So given the weight and the height, we classify as healthy or overweight or of high risk for diabetes or what have you. So the process may look like this. This goes all the way back to the classic text by Duda and Hart. This is back in 1973. And here maybe our data, the healthy are the green, the overweight are the red, they're higher on the weight, lower on the height scale. And we assume that some expert has labeled the subjects as healthy or overweight. We have some clinical diagnosis. There's a reason why that is. And based on that, we may learn a line. The line shown that separates this thing, that is the classifier. And when we get a new measurement, if it's above the line, we say, okay, that's healthy. And if it's below the line, we say that's high risk or overweight or what have you. That is the most fundamental part of this process. Now, this is one way to do it. Another algorithm that, again, dates back to that era is the nearest neighbor algorithm. And here we memorize all our examples. So this, the old saying in AI is that memory is a substitute for intelligence. And when we get a new example, we classify based on what is nearest neighbor or neighbors where. So if you do that, you get an implicit boundary that looks like this. Below the curve, you're in the red category. Above the curve, you're in the green category. And that's just driven by how close you are to an existing sample. If we were to get one more sample up there, maybe now we have a three-part partition. Top and bottom are high risk, middle is low risk. And this is either the nearest neighbor, or we have K nearest neighbor, or K can be 5, 7, 9, depending on your application. Let's continue the story. You could have a situation that looks like this. And now maybe get another example. And now we have an island in the middle. So this little spot here with the red star is in the middle of the greens. Now inside the island, your high risk, below your high risk, above your high risk. And this type of structure is more common when you look at deep neural networks. You have a lot of islands, really. You don't have a clean separation. What happens at those points labeled with question marks that are far away? What happens if a doctor sees a patient that's unlike anybody they've seen before? The answer is that their experiences are going to help them. Same happens to classifiers. And coverage is critical, okay? We're unlikely to get good results if we're far away from training samples. If our patient doesn't look like any patient we've seen before, whether visually, we have a patient of a different skin color than our database, or the presentation of the disease is very different, we're unlikely to get a good result. And that is the fundamental limit of all machine learning, deep learning. We need a lot of samples and we need good coverage. And we'll come back to that again. Here is what a digital neuron looks like. This is the building block for a neural network. We have an input layer, this numbers X1, X2, and 1. So the inputs, we have some weights, W's and the B here, and we have an output Y, and this is the Y is the simple neuron, and the neuron could have an output like a function that's sort of a function of the weighted sum of the inputs, so X1 times W1 plus X2 times W2 plus B. So we have the input layer. This is what we feed into the neural network, the weights and the output. And then in practice, what happens, we give it lots of examples of X's. We know what the Y's are to be, and we tweak the weights to get the right answer. And that is how you trained a cell. The function F may be something like a threshold potentially. If A, what goes into F is greater than 0, we can output 1, otherwise it's 0, and we say the neuron fires by the biological analogy. There are other forms of function available, ReLU, sigmoids, what have you. And again, just to reemphasize, we learned here the weights, W1, W2B, from example data, and then we apply them to a new input. Now, if you take lots and lots of these things, that's a final point here before I'm on the next slide. I'm just going to highlight that thresholding process if A is greater than 0. What we have here is an example of a nonlinearity. If we go from A minus 0.001, we get a 0 to A 0.001, we get a 1, right? So a small change in the input can give a large change in the output, and we'll come back to that. So if you take a lot of these neurons and we clone them together in parallel to create layers, so we have lots of layers one after the other, We get neural networks, and if we have a large number of layers, we get deep neural networks. That's really all it means. It means you have a lot of layers. To train this, we need lots of examples. We guess the initial weights and adjust them so the network learns the correct answers. This is a process known as backpropagation, more neural networks. It turns out in imaging, not by design, but that's how life works, that the first few layers learn features, they learn a representation, and the last few layers learn a classification. The power of deep neural networks and why we get such good results is this integration of feature extraction and classification. So instead of trying to figure out by hand what the features ought to be, the weight and the height, we give it as much as possible the raw data, the images, and it figures out what the right features are and then uses those to do classification. And so you can get features that are optimal for separating classes as opposed to features that are optimal for something else. How do we evaluate this thing? So here is the typical process. We need a separate training and testing set. That's probably the single most important thing. That flowchart above shows the process. We have some training data. We use it to train a model. We have a separate set of testing data, and we use it to test the model. And the reason for it is, especially when it comes to deep learning, these are so powerful that they can memorize the training data. So we only really are interested in how well does this model generalize, how it well performs on data that it was not trained on. Otherwise, we can just memorize the data. Ideally, we should use a completely separate testing set. In practice, most applications, because we don't have as much data, we separate at the start of the process. We take our data, we split it off into testing and training. We do the training and the testing. And you can do permutations of that, cross-validation, what have you in some cases, but we'll leave that for later. The fundamental problem, as alluded to before, is overlearning. It's a great cartoon that illustrates the case, so let's just walk through it. The title says below, thanks to machine learning algorithms, the robot apocalypse was short-lived. So Soldier 1 asks, this was surprisingly easy. How come the robotic uprising used spears and rocks instead of missiles and lasers? And here comes the answer. If you look at historical data, the vast majority of battle winners use pre-modern weaponry, right? History goes back thousands of years, and most of those battles were fought with stick and stones. To me, if you want to put some math on it, the statement, a measurement, like what happened in a battle, consists of perhaps the generalizable information, what you should take away from it. Larger armies tend to win. Something that was idiosyncratic that just happened to happen at that event, but doesn't really something you want to carry away. You know, if you're tagging in the morning, you tend to win, maybe, maybe not, but it may have happened a few times and you learn it. And of course, noise, the report we have of the battle may not be exactly what happened. It may have been altered to protect the reputation of winning generals, emperors, etc. The goal of machine learning is out of our measurements to learn something that reflects the generalizable information and hopefully filter out the rest. Easier said than done, but in general, the generalizable information is the most stable. So if we don't try to overlearn from our data, to not overfeed our data, we'll extract that. And as you try to squeeze more out of your data, you end up in the second and third category. The training data is critical. So the last military adage of the day, this quote is attributed to many people, so I can't claim to figure out who it is, but it says amateurs talk strategy, professionals talk logistics. I would say in machine learning, amateurs talk networks or structures, professionals talk training data. Not that the networks are important, strategy is important too, but training data is often more important. In machine learning, the code is often standard, it's often the same networks. The unit is the most common image analysis network, it's fairly commonly used. What often drives the results is the data, not the code. If you think about overlearning or overfitting, back to Den Haag, back to 50 years ago, here's an example from the book that it's the same problem that we're still dealing with 50 years later. We have some measurements, those are maybe our yellow dots. We try to extract some patterns, so maybe we feed a line, a linear regression, and has that sort of thing, and we have learned something about the data. Maybe we think there's a little bit more we can squeeze out. Maybe with that parabola, this green curve, so it goes up and down, so who knows, maybe there's a better fit. If you go to extreme, you can fit a super highly nonlinear polynomial, and this fits our training data exactly. But the question, of course, isn't how well you fit your training data, these measurements, but what happens when you get another measurement there, where the blue point is? So the prediction error may be vastly different between the three. And in general, if you overfit the data, you get large generalization errors compared to fitting errors at the beginning. So keep that picture in mind. We'll come back to that as well. So as an aside, just from a research perspective, there's also here the danger of reusing your data sets too many times. So what I said we do, we train and We do that process once, we train our neural network, we test it on the data, we did it once, so far so good, everything is clean. The results are not good enough, so we learn another model and another model and another model. And even though we don't mix training and testing data, implicitly we add. We get feedback from the testing data into a training set. And training multiple models can lead to overlearning from the data. And this is the same problem if we do this community-wide. It's a form of overlearning. We train lots of nonlinear systems, some of which are likely to work by chance. So back to my nonlinear case, if 50 labs train a nonlinear system on this data, and we've got 50 high-order polynomials, by chance, one of them will generalize. But are we learning something about the data, or did we just get lucky? Did we do? And in the social sciences, this form of training lots of models on the data and not doing proper comparison correction led to a crisis called p-hacking. And I wonder if in the machine learning, we're going to end up in a community-wide p-hacking. Are we really learning something? Or if 100 groups train on the same data, one of them just gets lucky. And as an aside to that, this is where we get into trouble with constant reuse of the same data sets. By the time the 5,000 paper or the 10,000 paper is published on a data set, whether it's the Human Connectome Project or ATNI or Abide or all of those things, are we studying the brain at this point, or are we studying the data set? So something to keep in mind. And we'll close that parenthesis here. Related to the problem of learning is the fact that deep neural networks are highly nonlinear. The picture you see on the left is the cover of this book by Edward Lorenz, The Essence of Chaos. Lorenz is sort of the father of chaos theory. And the summary of nonlinear systems is that small changes in the input can lead to large changes in the outputs. Back to the cover is the famous butterfly, a butterfly flapping its wings in the Amazon can lead to a tornado in Texas. This is sort of the summary of highly nonlinear systems. And if you look at deep neural networks, each layer has activation functions, which are nonlinear. There's a threshold, a ReLU, what have you. And we may have 100 layers. So there's a ton of linearities, you add them one after the other. And the expectation, if we knew nothing else, is that these networks are going to be sensitive to small changes in the input. Excuse me. Small changes in the input data will lead to large changes in the results, and we're going to be in this domain. Do we see that? And the answer, of course, is yes, and I'm going to show you some examples. This is a first example. This is from an interview published in the Canadian Healthcare Technology Journal earlier this year. And I'm going to quote from from Dr. Nadma Dabushi, one of the well-respected colleague, now Georgia Tech. At the time, he was at Case Western. This work was done. I actually heard this story from him personally when he visited Yale about the time this happened. He said in 2018, a team at Case Western, where he used to be, developed an algorithm that looks at endomyocardial biopsies using slide images to determine whether patients had evidence of heart failure. And they trained a deep learning network, and it was 97% accurate, was significantly higher than the humans they had at the time. So they got really excited. Sometime afterwards, they obtained another set of cardiac images, and they ran the same algorithm. And strangely, they got an accuracy all the way now down to 75%. So what happened? It turned out that between the first and second batch of testing, there was an automated software upgrade to the slide scanner, and that changed the images enough to affect the way the algorithm interpreted the images. And this is his conclusion. It was a case of AI drift, small changes in the input, small changes in the data. And in Dr. Madabushi's opinion, it's a cautionary tale. You have to stop trusting the black box for high-stake decisions. You have to be careful. So here's a case where the only thing that happened was a minor upgrade to the scanner. It was enough to destroy this algorithm. And that's because probably in the training, they learn not only how the disease appears, but how it appears in the specific configuration, the specific software, the specific scanner. We made a small change of that, and the overlearning came through, and that's what they call drift. Here's another example from our own work. We're trying to segment prostate MRI data. The top is a set of data we got from our colleagues at Stanford. The more important part is from a GE scanner. The bottom is some sort of prostate data we got from our own data set at Yale. This is on a Siemens machine. And we trained a neural network to do this segmentation, and we followed the right things. We did threefold cross-validation. We trained from a single site, and we did testing either from the same side or another side. So if you trained on Stanford data, tested on Stanford data, or trained on Yale data, tested on Yale data, got really good results. Accuracy, the median accuracy in the high 90s. If you trained on Yale data, tested at Stanford, we have some deterioration, but still pretty good results. But the kicker was this final one. When we trained on Stanford data, tested on Yale data, most of the subjects failed. We had the median accuracy was close to 0. And again, this was a case of the images were different enough from the two scanners that we failed to see that. And the lesson I took from this is that you can't really say anything reliable if all your data comes from a single source. You have to do more sources. We're able to correct this by doing intention normalization. It's a fairly easy problem as things go because of obvious failure, but keep that in mind. Let me give you another example from outside the imaging world, and this is the sepsis algorithm that's part of EPIC, the electronic health record database, and this was their proprietary sepsis prediction model. This is one of the major issues in hospitals, predicting which of the patients are at risk for sepsis and taking measures. So a set of researchers at the University of Michigan, you can see the team Wong, Otles, and Donnelly, repeated the study with their own data, and in a cohort of 27,000 patients undergoing about 39,000 hospitalizations, they had 7% occurrence of sepsis. The EPIC sepsis model predicted the onset of sepsis with an area under the curve of 0.63, which is substantially worse than the performance reported by its developer. The study suggests that this model poorly predicts sepsis, and it was widely used at the time despite poor performance, and raises fundamental concerns about sepsis management on a national level. They had to recall this model and retrain it. Now, in the original work, the area under the curve was on the order of 0.8. They got about 0.63. For those of you who don't think in area under the curves, at a very hand-wave approximation, an area under the curve about 0.8 is the equivalent of a sensitivity and specificity of about 0.8. 0.6, you can get that with a sensitivity and specificity of 0.6. There are other ways to get the same area under the curve. But think about it as a 20% reduction in both sensitivity and specificity. So it's pretty dramatic. One additional example that's kind of interesting is work from Sue Vargas and Guichi from the computer vision world. This is the one pixel attack work for fully neural networks. What you see here are images, and these were trained on a classifier, and they were getting the correct labels. And then what they went ahead and did is that they changed a single pixel in this image. For example, in the image, the second on the top, they added that orange pixel on top of the tire, and that was enough to fool a pre-trained classifier to report that this was a dog instead of a car. And they were able to do it with any pair of images to get any label to any label by just changing one pixel. And this again is a case of the high nonlinearity in these networks. Highly nonlinear networks, susceptible to changes in the input, and they were able to demonstrate there was a single pixel attack on all these things. One final example before we try to make sense of it, and this comes from functional magnetic resonance imaging, some published work from my colleagues at Yale, Matt Rosenblatt and Dustin Shinos. What you see above at the top is a connectome. This is data derived from fMRI by averaging a parcellation of the brain, and then they use this to predict behavioral measures outside the scanner. So can you predict IQ from fMRI against measured IQ? And you can see in this particular data set, the prediction was not that great there. Correlation was about 0.18. They went ahead and they added a little bit of enhancement patterns, targeted enhancement patterns to the data. These are very small compared to the original data. You can see the correlations, those 0.99s. It's how similar the corrupted data were to the original data. But that small data, you can think of it as almost fraud, right? They're just showing how sensitive these algorithms are. Raised the IQ prediction from 0.18 to 0.46 all the way to 0.97, which is a tiny change. And again, this is not a deep neural network, but it involves a significant number of nonlinearities stacked together, small changes in the input, large changes in the output. Why do these things work? Why do adversarial attacks work? If you train a neural network, perhaps those are your data. The green dot are your healthy controls. The orange dots are your patients. The problem is that in the big empty space of all possibilities, all we know is the truth at the location of those points. So yeah, we think we may get a result like this, nicely far away from everything. And that's what happens when you run a linear classifier. You can specify how far away you can get from data. And so you're somewhat robust. But with a deep neural network, you may well end up with a classifier like this. This has equivalent truth value than the other one, right? All the patients are on one side, all the controls are on the other side. The problem with the second classifier is it takes very little movement of the arrow at the top to shift it from one side of the classifier to the other and to get an adversarial attack or to get an error. And again, this is a function of the nonlinearity you see. One solution to this problem is augmentation. So you take your initial data points, you see them on the right, and you make them fatter by creating more examples from the existing samples, right? They smooth, you change the contrast, we rotate, we deform the images, we generate more examples, we can use synthetic generators. And, just at a high level, think of it as taking little dots and making them fatter, and this gives you more points. So this can add stability, and it actually, in fact, does work quite nicely. But are we sure that the created synthetic samples maintain the same level? Did the change actually make it more like the other class? If we take a healthy subject and change its image, is it more like a patient now? Is it still a healthy subject? Just to give you a couple of examples of what happens, we all learn deep learning on the digit data set, the digit recognition. If you rotate a 6, you get a 9, so you change a class. A more sophisticated example, perhaps, let's take smoothing. If you look at epilepsy on the gray-white boundary, one of the structural abnormalities of epilepsy is cortical dysplasia, and this manifests sometimes as a blurry gray-white boundary. So if you smooth normal tissue, you actually get something that looks like a cortical dysplasia. So augmentation is helpful, but it's not perfect, and we need to be careful about that as well. So we've seen some of the issues. We know that we need to follow appropriate processes and your risk classification and your all of that. But let's look at a couple more things from the regulatory process. This is the last major chunk of the talk. We'll talk about how to evaluate to establish trust, and we'll talk a little bit about managing the risk. Can we live with the mistakes? Maybe we can in some cases, and sometimes that's the easiest thing to do. One of the issues with machine learning systems is all we can really test is a complete system. Unlike regular software, we can check all the components and test function by function and make sure every function works and then put them together. In deep learning, all we have is like the black box, input, output. So given the series of that, we really need to make sure that our testing data is diverse and covers the true variability of the population, right? We don't just test on a subset of the population. The old story about drug studies only been done on college undergraduates and not working on older people applies twice as hard here because of the nonlinearities involved. Ideally, you want to use data to test that the development has never seen. If it's a serious high-risk operation, maybe this is the case for prospective data or separately data from another location. And this is hard for us in research, but we have to realize that the validation is almost never finished. We need to follow up and monitor in production. And you can see some of the echoes of the regulatory process there. One of the documents the FDA has, this now dates, 6, 7 years, depending on when you count the drafts. This is one of the evaluation documents for software as a medical device. This precedes the deep learning era, but the ideas are still valuable. So if you want to see 3 things as part of your evaluation, the first one is to establish what's called valid clinical association. And so crudely spoken, is what you're proposing plausible? Are you measuring something that's somewhat hopeful for this disease? We'll get an example in a minute. Is what you're measuring accurate? This is the analytical validation. And then the third component of that is the clinical validation. Does your measure make a difference? So is it plausible? Is it accurate? Does it make a difference? So let's look at an example. Let's take, again, a traditional algorithm. We're doing tumor segmentation. So what do these things look like? Well, the valid association may simply be a literature review to establish that tumor volume is a useful predictor in managing the disease. Small tumors, manageable, large tumors, highly risky, etc. That type of thing. Your analytical validation may involve comparing the output against some gold standard to ensure that your algorithm is accurate, that your segmentation result is correct. And your clinical validation may mean apply this method to a new data and see if this measurement is helpful in stratifying patients. Just because you can get it accurately and just because you measure something that's relevant doesn't mean it's actually useful in a clinical sense. Finally, if we get out of this document and go back to the rest of the guidance, we have to worry about safety. Safety means in software mostly usability, that we don't cause the doctor to make a mistake. So perhaps in this case, we would allow the radiologist to look at our overlay of the tumor on top of the image and check that it's accurate so they know whether to trust it or not. So this is what we think the tumor is. This is the image. You look at them together, you can say, yes, the algorithm worked, no, it didn't work. Now let's look at this one step up now in the context of AI decision-making. Is it plausible? Now, do we believe that there's enough information in the image for an algorithm to make this kind of decision? Is appearance from the image enough? Maybe the same thing could be very different depending on the underlying genetics. So it may not be plausible or not, but we're trying to squeeze more. Do we have a large and diverse training data set to train this model? And if we don't, now can we really predict disease outcome from 20 patients? That's questionable. Analytical validation is harder because we cannot do the detailed testing. We can check only the final result, so we're a little bit more limited here. The clinical evaluation is same as before, but given the relative lack of evaluation, it's more challenging to establish trust here. And safety, how do we evaluate a black box? The algorithm tells you that this patient is at high risk of getting Alzheimer's. Deep down, this came out of a black box. We may be able to examine what it did, but not in the same way as something else. Well, we tell you the tumor is here and it's here, right? No human knows how to do that. The final part of this talk, before we get into discussion, we'll talk a little bit about the risk management process and how we manage risk. Risk is something that goes bad and how to control it, how to make sure that if a bad thing happens, it doesn't has downstream effects on our patients. In machine learning, we can do this at 3 levels. We can control the inputs to our software AI. We can eliminate problems before they go in. Perhaps we realize that this image is not like any other image we've trained in, so we can just issue a warning, right? Our algorithm is unlikely to work here. It's from the wrong scanner, the wrong resolution, the wrong contrast, what have you. You can do things to make the system, the deep learning neural network, less sensitive. There are training methods. I think that's less hopeful. And you can provide checks at the end of the process. And this comes under the rubric of explainability. Can we provide some explanation to our user as to what the network did so they can evaluate the result? My colleague Christian Kastner, Carnegie Mellon, always talks about one should think about AI modules as unsafe components in a system and build checks around it. And these checks have to be almost non-AI, things that we can fully trust. So we can manage the process, we can build checks and balances outside the neural network to detect if something is going wrong and so hopefully some warnings. What could go wrong? Can we spot it? The biggest challenge, if we're relying on humans as a risk management strategy, human in the loop type approaches, can a human even tell what the results should be? And this is easier when we're automating something a human can do, something like segmentation. So this is where, a particular part of the structure of the brain is in the image. This is where the tumor is in the image, because a human could theoretically do that. It would be slower, it would be perhaps less accurate, but they can certainly check. If we're doing something like prognosis for future outcome of a disease, that's a much harder problem because humans already can't do that. So that's a harder thing to do. Can we explain the result? Now, given this super highly nonlinear neural network, can we really come up with a number that helps you see what it did? That's a questionable thing. And in the literature, there's a whole debate about the bias of trusting algorithms or distrusting algorithms. Some people believe computers more. If a computer came up with the results, they believe it more. Others implicitly distrust computers if the result came from computer. And the literature is a little conflicted here, but it's an open problem, as you can tell. So let's look at one example of explainability for imaging applications. This is an example of a neural network that was trained to detect some particular kind of lung disease. And you can interrogate the network to see what parts of the image play the most role in its decision. In this particular case, it was looking on the shoulder. So that's in a case of, this is what's known as a heat map or a salience map. And you can tell by looking at this that the neural network is looking at the wrong place, so probably shouldn't trust it. So the goal is to provide understanding to the user, and sometimes it's very useful. So in the case on the left, given that the algorithm is looking at the shoulder and not on the lung, probably you shouldn't trust it for a diagnosis of lung disease. But can we really summarize a high-dimensional nonlinear system with millions of parameters into a single image map? Sometimes we can and sometimes we can't. And the other problem that comes up with explainability is this sort of aphorism in Greek is very famous. Some knowledge is sometimes worse than complete ignorance, and this is the illusion of understanding. So what if an algorithm showed that it was looking at the lung? It doesn't really mean that it interpreted the image correctly. It means that it was only looking at the right place. So if you had the explainability and it told you it was looking at the right place, maybe you'd be more inclined to believe it, but for no good reason sometimes. So these are some of the challenges of explainability. So I don't think that's going to be the panacea at the end of the road here. Just as one more slide on explainability, there was a recent study where they did benchmarking of methods for chest X-ray interpretation. And this is from their abstract. Salience methods produce heat maps that highlight the areas of the medical image that influence model prediction are often presented to clinicians as an aid in diagnostic decision-making. These people tested 7 methods, and while one did better than the others, for our purpose at least, all 7 performed significantly worse compared to the human benchmark. And their work demonstrates, and it's a very interesting paper, that several important limitations of salience methods must be addressed before we can rely on them for deep learning explainability in medical imaging. So explainability is important when it works, but can we trust that either? So just to wrap up the talk, I have about 4 more slides. So I think we're okay time-wise here. The first question that sometimes to ask when you say, can we trust this thing, is whether the output is the tool or the result. If we have a tool that helps you diagnose cancer or Alzheimer's from an image, here the tool is the result. So we have to learn to trust the tool, and it's a more difficult process. If the tool is used to give you a lead into something else, to identify potential drug molecules or some interesting work in that area, which you then test separately, then maybe trusting the tool is less important because the actual outcome isn't the tool, but the tool gives you some input into some other process. Can we afford to be wrong? What if we got it wrong? Can we mitigate or manage the risk? In some cases we can, in some cases we can't, and that will determine what happens. Back to my sort of take-home slide, it's not what the tool does, but what it will be used for. So let me give you 3 examples. Those of you who are Star Wars fans may have heard that they use deep learning to generate some deep fakes for one of the recent Star Wars movies. Deep learning used to generate movie sequences can fail 99% of the time. That's not a problem. You throw away all the bad results, you keep the good ones, and so long as you did it in a reasonable amount of time, you can use it and you're happy. Perhaps closer to medical, if you had thousands of images for a clinical trial or a giant study and use an AI tool to process them, you might be fine with a decent failure rate and not require a human to check it, because maybe the error average is out, so long as the tool is mostly accurate. On the other hand, if you're using the same tool to diagnose individual patients, you may have a problem, right? So it's what you will use it for. It may be the second algorithm that does 2 and 3 above the clinical trial or the individual patient. You trust it for one, you don't trust it for the other. So it's not a question of can we trust AI, it's when can we trust it and when can we not? That is the question to keep in mind. So you've seen all these AI chat things. This is from ChatGPT. So I went ahead and asked, what are the main issues of the use of AI for medical software? And it gives a very good answer. If my students gave me that, I might actually believe them. So that's now I'm scared when it comes to teaching next semester. But what I want you to focus on is the statement here. Because AI systems are only as good as the data they're trained on, any biases or errors in the training data can be amplified in the prediction made by the AI system. This can lead to incorrect diagnosis. Treatment recommendations can have serious consequences for patients, and this includes fundamental issues like bias. And so let's just touch on that briefly. I alluded to earlier in the talk, but let's just talk about it quickly. The training data can be the root cause for bias. A particular group may not be very well represented in the training set, so you get less accurate results there. This is not always the case. Sometimes a larger but a biased training set can give you better results for all groups than a more balanced training set, but that's smaller. So there is shared variance across groups. And so sometimes a larger biased training set may be better for everybody. So that's just something to keep in mind. I think, to me at least, the key issue here is that we must be able to provide fair warning that our results are less accurate in a particular group, which means that we need to separately quantify performance in different population groups. We'll never be able to do the same performance in everybody. Sometimes the anatomy of different people make it easier to diagnose a disease. Another skin color may make it easier to diagnose a particular skin disease on another. But at least we need to know whether this algorithm works differently in different groups. That's sort of my take here. Finally, we talked about AI, but let's think a little bit about software generically and research. Some of the issues involving AI apply to all software as well. AI is always really a component of a software package. Can we trust research results that depend heavily on code? What would you look for? So I would suggest that we don't just look at the results. We should ask questions like, who did the programming? How well trained were they? How well was the code tested? Did they have quality systems in place? Are they suffering from essentially the same thing in AI, as in what we call an AI distribution shift? Did they take code from another lab, they used it on their own data, and their own data is slightly different, and they trusted the results? So those are questions. So as a final slide, just to highlight this change of use case, this is a tweet from Neil Ferguson of the Senior Epidemiologist in London, and they were using some of his code to predict how the different control measures for COVID would come out. The tweet is from March 22, 2020, so you can tell the date. And at the time, and this is in response to people wanting to look at the code that they were running, and he said, look, the reason this is being delayed is that I wrote this code, and this consists of thousands of lines of undocumented C, 13 plus years ago to model flu pandemic, so it's taking me a little bit more time to get it out to you. Now, this code may have been perfectly fine as a modeling tool for research, but is it fine as a tool to sort of do public policy? So studies should be used with care. Just as from a previous slide here, high risk requires more detailed documentation. So you can't just use code that was designed for one purpose for another purpose. It may be the same tool. It may superficially be the same purpose when modeling a pandemic, but the use of this code is very different. One is to teach, perhaps. The other is to decide what you should do. So this with no criticism of what they did, in fact, there. That's not the issue point I'm trying to make. With that, let me acknowledge funding here from the NIH and the NSF. And I'm not going to list all the people because I've had many discussions with many people. I'm worried I'm going to miss somebody and feel bad about it. But all the people who have heard practice versions of this talk and these ideas from me over the years, I'd like to thank you all, and you know who you are. Finally, for those of you who want to know more on the medical software side of this research, there's an online Coursera class on medical software that you can take. The class is free. It's a small fee for the certificate. And you can hear me talk for many more hours. There's a companion textbook that we wrote that I use for my own teaching that was just made available. And you can get more information at the link at the bottom, medsoftbook.com. And with that, Leonid, we'll stop here and we'll take questions.